In mammals, such as mice and humans, a small subregion of the hypothalamus called the suprachiasmatic nucleus, abbreviated SCN, serves as the master biological clock of the animal. Neurons within the SCN display feedback loops in the expression of certain proteins. Let us begin by looking at two proteins, named clock and bmol1, that are expressed at the beginning of the day in these cells. bmol1 and clock bind together to form a dimer and enter the nucleus. Note that in the nucleus, the genes that encode these proteins are active. They continue to be transcribed into mRNA, which is exported from the nucleus for translation into bmol1 and clock proteins. The dimer binds to the cell's DNA at regulatory sequences called E-boxes. A number of genes contain E-boxes. Several important ones include reverb alpha, cry for cryptochrome, CCG for clock-controlled genes, and PER for period. Several PER genes exist in mammals, but the function of only one PER gene, PER2, is clear at present. The binding of the clock bmol1 dimer to the E-boxes of these genes promotes transcription. The mRNA molecules enter the cytoplasm, where the RNA is translated into proteins. The protein functions are diverse. Although protein from a single clock-controlled gene is shown here, a variety of clock-controlled genes are expressed in this manner, and it is the CCG proteins that control aspects of the body's daily metabolic states. Reverb alpha is an important protein that negatively regulates bmol1 and clock expression. Recall that when bmol1 and clock proteins are expressed, they turn on E-box-containing genes, including reverb alpha. Thus, the action of reverb alpha ultimately results in the reduction of reverb alpha expression. Meanwhile, PER and cry proteins bind to each other, and the complex enters the nucleus. The complex binds to the clock bmol1 dimers and inhibits their function. Thus, PER and cry inhibit the transcription of their own genes in a classic negative feedback loop. The mRNA previously transcribed soon degrades and no new proteins are made for a while. Because the previously made proteins also degrade with time, eventually the inhibition due to PER and cry is lifted, starting the whole cycle over again. This molecular cycle, which takes about 24 hours to complete, drives the 24-hour activity cycle of SCN cells. However, other factors synchronize or entrain the molecular clock to the day-night cycle. For example, certain retinal ganglion cells detect light in the environment with a light-sensitive pigment, melanopsin. These retinal cells project to the SCN and provide cues via the neurotransmitter glutamate about day and night. The glutamate promotes a chain of events that leads to an increased transcription of the PER gene. When the animal's photoperiod is shifted, this light-mediated boosting of PER production shifts the phase of the molecular clock and therefore the animal's behavior.